Dr. Gregory Abrams, thanks so much for coming on to Evolution Soup all the way from your office in your home in Belgium. You are an archaeologist and postdoctoral researcher at Ghent Archaeological Science Centre, Ghent University, Belgium, and your main focus is on the archaeology of Neanderthal sites in Belgium and Northwest Europe. Today we'll be exploring not only the intriguing fossil bone tools found at the cave sites in those areas, but also looking into proteomics, an exciting new technology that goes where DNA testing cannot. Welcome to the show, Greg. Well, you've been studying these Neanderthal caves of Northwest Europe for almost 20 years. These new findings must have been a welcome surprise.
Hello, Mark, and thank you for asking me to be part of your channel, your YouTube channel. So it was a very interesting and exciting discovery we've made just a few months ago about some tools made from cave lion in Scladina cave in Belgium. And so to assert the identification of these tools, we use very, I would say, outstanding research and techniques such as the proteomics. And thanks to that, we were able to identify very clearly the species that have been used by Neanderthals. And as it is the first one, the first discovery of that kind in the world, it's very interesting, and it raises more questions that it provides answers.
Before we talk about these Neanderthal caves and the tools found within them, let's just hear a little bit about your background. Greg, where did you grow up, and what were the things that fascinated you and led you to the work that you're doing now?
So I made my studies in Belgium for archaeology, and I had, a couple of weeks ago, I met my old friends from primary school. So when I was between 6 and 12 years old, and basically at that time, I was already interested in history and archaeology. And so during all my secondary studies, I was interested in being a professor of history for secondary schools. And when I started the university, I was in the field in Scladina cave, to be honest, for instance, and I just fell in love with the field archaeology. And so I did everything to be a specialist in prehistory. It's a very long history for me, this idea of being an archaeologist. So as far as I remember, I was always interested in the past of the human and the mankind.
Well, Greg, you've been studying the Neanderthal cave sites of Northwest Europe for almost 20 years, as we said. So, what can you tell us about these locations, as well as the early discoveries made within them?
So, as you maybe know, Belgium has a very long history in research about the Neanderthals. So, if we are going back to 1829, it's the time where we have found the very first Neanderthal remain. It was in a cave in the Meuse Valley named Angis. And unfortunately for us, this Neanderthal child has not been recognized as a Neanderthal directly. So we had to wait almost a century to identify him. If we just continue the 19th century, we see that we have also discovered many other sites, such as the Spee Cave, which is a very famous one, where we have found for the very first time some Neanderthal remains in context associated on the very same stratigraphic layer with some lithic tools and some faunal remains. And so basically at that time, it was already known as a big discovery, a huge one. And since then, we performed a lot of excavations in different sites and in different caves during the end of the 19th century and all the 20th century. And it led us to 1971, when we have found the Scladina cave. And this site was not touched, so it was almost completely preserved from previous excavations when scientists started to work there. And we found in the early 80s what was called and understood as something intact. So a kind of, the ground where the Neanderthals stood and where they made all the activities inside the cave. It was, I mean, with the time that passed and the interpretation and the knowledge we have now, we can assert that it was not the case. It's not an intact floor, an intact ground. Anyway, everything is very well preserved. And it's in this layer, which is called the Unit 5, and which is dated from the end of the Salians. So it's older than 130,000 years, that we have found a lot of faunal remains with anthropogenic modifications. And during the 90s, research has been made on that material, and they found that these Neanderthals maybe used that cave as a kind of hunting camp, a short-term hunting camp for chamois. We have found something like 14,000 lithic artifacts there, and a lot of chamois remains. And I would say until just a decade ago, the identification of the remains and the faunal remains, they were made by, I would say, classical anatomic research, by comparing these material with modern species and with reference collections. It's easier to make that kind of identification using the shape, the morphology of the remains when they are almost complete. And the issue with the material that has been modified by the Neanderthals or by human or by, I would say, even carnivores, it's that the material is reduced, is very highly fragmented. And so it's more and more difficult to identify this kind of fragments when they are almost, I would say, so broken that there is no landmarks that can help us to identify clearly the species. And so we had, when we look at the first publications, we had a lot of material that has been identified, but most of them were what we call the unidentifiable fraction. And this material is the one which was the richest for anthropogenic modifications. And so, as I said, until a decade ago, it was barely impossible to make this kind of identification. And if we go back to the cave bears, the first fragment, the first bone tools I identified in Scladina were made on these cave bears, and they were fragmented, but highly fragmented. But I spent a lot of time refitting the material together. And so basically, by sticking a fragment on the other and so on, I succeeded to reconstruct an almost complete diaphysis, a shaft of a femur. And based on that, we already showed that it was possible to have more information about the Neanderthal and the Neanderthals and the way they processed the carcasses of these very large carnivores. So I did what the archaeologists did on the lithic material. I did a 3D puzzle with these remains. When I try to explain the importance of doing that kind of thing, I take as an example the David of Michelangelo. And we can, when we are in the museum, we can look at the statue and see how beautiful it is. But we don't have any evidence, any piece of evidence of how Michelangelo sculpted this statue. And so, if by any chance we could discover all the fragments of marble resulting from the creation of this statue, we would be able to see if he started by maybe working on the head, on the elbow, on the ankle, and I mean whatever. But we would be able to look at the process, the production, what we call the chaîne opératoire, that was used by Michelangelo. And this is what we are doing in archaeology. So basically, all the material that could be exported by Neanderthals, they brought it elsewhere, but the remains that were lying on the ground, it's the remains that were not useful anymore. And so it's why by refitting all the things together, putting all the different pieces together, we are able to estimate what is missing, but also how did they produce these tools.
Your most recent research has made some astonishing discoveries regarding the bone tools carved and recarved by Neanderthals in a site called Scladina Cave in Belgium. So, tell us about these findings.
Yeah, we started this work something like 15 years ago in the cave, and Scladina, as I said, is a very interesting cave because most of the things were almost intact when the official excavation started, and especially for the Salian layer. That said, I started working on these very small fragments of bones, and when I was looking on the surface, basically I was doing what we call a taphonomic analysis, meaning that from the time of the deposition until the time of the discovery, many phenomenon occurred and affected the preservation of the faunal material and the remains. And so, reading the bones as you can read a book, we can go back in time and see the succession of these alterations and see what was going on and what was the aim of these Neanderthals when they modified those remains. And so I found some of them, and some of them by refitting I was able to identify that they were cave bears. And one of them was very strange at that time. I showed it to a colleague from the University of Lille, Patrick Auguste, a paleontologist, and even him said that it could be a cave bear, but something was strange on this fragment of tibia. And we said, but we can go further because it's something that could occur on an individual. So it's not something related to the species, but it could be something more related to the individual. And so it's what we call the variability of these individuals. And we did some analysis, and we succeeded to identify these as cave lions. And as I did for the cave bear, I made some refitting with these bone tools. And the thing that was completely crazy is that they have a kind of there was a kind of very strange breakage on one of the surface, and this breakage is very similar to those I observed on the lithic material. And so when you look at that, but not from, I would say, like a zoologist, but more like a technologist and an archaeologist, you see that there were very strange features, very strange marks on these bones. And when I did the refitting, I thought that we had a kind of flaking on the opposite of the breakage. So were very strange features, and these were possibly the marks, the modification that can occur when you use these bones as a chisel. And so, because this breakage and this pattern occurred before the use of these different fragments as bone retouchers, we were able to, at some point, recreate the history, the story of these tools. So, first of all, with all the marks that were present on the surface, we've seen that there was still some meat on the surface. So the Neanderthal removed the meat. They also scraped the surface to clean and to remove what we call the periosteum. So the bone was still very fresh when the Neanderthals used it and modified it, and then they firstly use it as a chisel, and I don't know why, but it broke during the use of this tool as a chisel. So the chisel has been probably used on the soft material such as wood, and maybe because it was already broken, they have decided to recover the different fragments to modify some lithic artifacts. And it's not the only thing, because it concerns two of the tools that were made from this single cave lion tibia. So it seems that for the other fragments, the purpose was already to create some bone retouchers. And so there is something, I would say, it has been made in purpose. They recover I would say, a fresh lion, or at least the tibia was very fresh when they recovered it, and they modified everything, every single bone to make them efficient. I mean, when you handle them, you see that it fits perfectly with your hand, and they have been used to modify the bones. And I can assert that because, as most of the bone literatures I studied so far in Scladina cave or elsewhere in Belgium, there is a kind of common pattern. It's the presence of very tiny lithic chips that are still embedded in the bone matrix. And so using some spectrometry techniques, the Raman or the EDX, so by looking at the raw material, we thought that it's something completely different than the bone matrix itself, which is the hydroxyapatite. And so basically, we identified plenty of siliceous material that were still embedded in the different bone retouchers, highlighting that they've been used as bone tools in purpose. And this kind of exceptional discovery, it's not the only one we have in Belgium. I said that we have a very long history of research in Belgium. I gave you some very small information about Angie and Spie, but there is also another site which is called the Cave of Goyer. And in this site, which is located 7 kilometers as crow fly from Scladina, my colleagues identified several Neanderthal remains, and on these remains, they have observed the very same things I observed on the cave lion and the cave bears and other species. So they use these bones, and they fragmented them, and to create bone tools.
Greg, you mentioned a new technological process called proteomics. How exactly does this process work, and why are its findings so significant?
So the proteomics, it's not a very new technique. I mean, it's something we are already using for almost a decade or at least maybe 20 years. This is, I would say, a very cost-efficient techniques to identify the material. When you modify the bones, when you break them, you create a lot of very small fragments, and using the common techniques of identification, basically relying on the shape of the bones, it's sometimes impossible and barely impossible to identify them clearly. So we have, and I would say the chemist and the biologist developed a lot of different techniques. The one which is the most known, it's the DNA analysis. The DNA is very fragile. The DNA is not all the time very well preserved, depending on the composition of the soil, of the humidity of the soil, of maybe the climatic fluctuation, and so on. And we needed something else to work with. And colleagues developed research and the identifications based on on proteins and proteomics, I would say. So it's a proteomic analysis, and this is kind of a groundbreaking discovery because the proteins that are present everywhere in the tissue. So when you are manipulating something, you put your DNA on the thing you are manipulating, but you also put some proteins. And the big advantage of these proteins is that they are less fragile, and they can be better preserved even for remains that are 700,000 years old, which is not possible so far for DNA. It's barely destructive, so you just make a little scratch on the surface of the bone. We just need 5 milligrams to make the identification, and using this, and by making a very strict protocol to dissolve the bone matrix, to recover the protein, the collagen, and so on, you can put the sample in a mass spec, and it will result with the identification of the different peptides. And by combining these different peptides, the mass of the peptides that have been identified and that have been recorded on the mass spec, you can easily, depending on many other factors, but you can, in a couple of days, I did it myself quite recently. So in a couple of days, I was able to make an identification of several dozens of bones. So it's very efficient in many ways. It's as I said, it's cost effective because it's much more cheaper than DNA analysis. And these are the big advantages. Sometimes you can reach the species for some of them, but most of the time you can only refer for the identification at the gender or the family level. Faunal remains and the identification of them in order to reconstruct the behavior and the exploitation pattern of these past species, it's more than enough. I would say, as I said, we the first in the first publication I wrote in 2014, one of the bone retouchers was identified as a cave bear, but thanks to the proteomic, it has been identified as a cave lion 10 years later. And it's something we are developing with two different labs. The lab from the University of Ghent, of Ghent and the other one from the University of Lille. We are working together in order to identify much species as we can in the collections, and to see that the idea we had a few years ago about the behavior and the way the Neanderthals exploited the species, it's evolving quite a lot.
So, how has this discovery changed the way we look at Neanderthals?
We used to think that Neanderthals were hunters and gatherers, and that they mostly hunted herbivores, even a little bit larger, so like bovids, horses, and deers, reindeers, red deers, fallow deers, depending on the time period. And it's basically what we have as results in most of the sites. And I was very surprised when I received some results from Proteomics about the cave lion, because it's the very first time we have bone tools made by Neanderthals on this species. And it opened so many possibilities about their behavior. Imagine that for the cave bears and the cave lions, the Neanderthal faced these large carnivores. So they were at some point in competition with the biggest predators that were living with them in northwestern Europe. And this is something completely, I would say, completely outstanding, because you can't avoid to and skip to compare the Neanderthal behaviors with the behaviors we observed in more recent periods. We think of Neanderthals, we picture them with non that developed weapons, with maybe not that developed strategies, and it's why at some points it's difficult for us to go in details in the interpretation or not. I'm a very basic archaeologist, and I rely on facts. And the thing is that when I look at the material and when I look at the modifications I observed on this tibia, I see and I'm sure that the carcass was fresh, because of the different marks we observed, the scraping marks, the cut marks, the breakage pattern that shows that this bone was still full of collagen when it has been broken. So it was still very flexible, and the way a fresh bone can break is highly related to the pattern is highly related to the amount of collagen that is still inside the bone. And so we know for sure that the carcass was still fresh when they exploited it. But that's it. Did they hunt this cave lion? Did they recover a dead one outside the cave and they just remove the leg to make their tools? I don't know. And at that point, you need to be creative and to imagine the thing. And if they recovered a dead lion when they removed the leg, when they recovered the leg, it was still fresh, so they were aware that it was a cave lion and a large predator. If they had to hunt it, I would say they knew more than us that this is a challenge for people to face the lion. It's not like modern hunters. You are in a jeep and you can shoot a lion from several 100 meters far from the lion. There, they to face it. It opens so many possibilities about the social organization, because You don't hunt that kind of large predators when you are not well organized.
It really is fascinating work that you and your team are doing, Greg, and I'm very grateful that you've been able to take the time to come onto the show today. So, what's next for you? Any new projects that you can tell us about?
As I said, Belgium has a very long history of research for the Middle Paleolithic and the Paleolithic in a general way. And even if we have found a lot of Neanderthal remains, we have identified so far 17 individuals spread in 9 caves. We have absolutely no remains related to the early modern humans, but we know that they were there because in one of my previous research that was related to my PhD, we dated early origination, so it's the culture related to modern humans, early modern humans, and we dated the origination, which was almost contemporaneous with the late Neanderthals, just a gap of a few centuries. And so we know that right after the Neanderthal disappeared from our region, the modern humans arrived, but we have absolutely no remains. And it's very difficult to understand why we have this lack of modern remains, because uh, when you look at the evidence we have of them, it's we have pendants made from ivory. We have also bone points made from reindeer antlers and so on, and they are quite well preserved. And so, if we had any remains that were obvious in the sites, we would have found them. And we did not so far. So we need to be a little bit more creative to think a little bit on the side, just to what can we do to find this kind of these pieces of evidence of their presence if there is nothing obvious. And it's what I'm doing at Ghent University now. It's We know that at that time the carnivores were present in the caves, and they were very active, especially the cave hyena. Identified 2 3 years ago a Neanderthal tooth in Scladina, in the deposits of Scladina, that has been digested by a carnivore. We have in another site, it's Font Forêt, a discovery made in 1898. We have found a fragment of shaft of a femur of Neanderthals, and we observed carnivore modifications on the surface. So we know for sure that they had some contact between carnivores and these late Neanderthals. And the question is, why not for the modern humans? If we have identified this just a few centuries or millennia before the arrival of modern humans, can we find this kind of thing for the modern humans? And if we find some, what what can we study and what can we analyze of these large carnivores? And my idea was the coprolites.
And for anyone who doesn't know, Greg, it's basically fossilized poo, isn't it?
Yeah, sure. It's the fossilized poo. It's quite strange, but these coprolites, it's a kind of I would say it's a kind of treasure chest of the past. Inside, you have all the things that have been ingested by the carnivore, because they eat it bones. It's very well preserved. So when you look inside them using the micro CT scans, you can see the different components of these coprolites, and it's full of plant remains, parasites, hairs, bone fragments, and so on. And what I'm developing now is to use different techniques to extract, I would say, virtually dissect the coprolites. It's the first technique, so using the micro CT scans. And the other one, it's to look at the components and on the proteins. And so it's why I'm working that much with the people from the different proteomic labs. It's because we need to be able to identify and to separate the different species that are present inside these coprolites. I'm closely collaborating with geneticists, with chemists, with biologists, and so on, to be able to take all the best tools we have to extract this information from this paleo poo. If we are lucky enough to find a fragment of early modern humans in these coprolites, we will be able to recover the DNA, the proteins, and so on, and to have a better idea. And I would say it's not a better idea; it's an idea of who were these early modern humans in northwestern Europe. I have colleagues from southern France for the radiocarbon dating, colleagues from Germany, the Max Planck Institute in Leipzig for the paleogenetic research, and colleagues from Ghent, from Lille for the proteomics. And so it's a big collaboration to have an idea of the first early modern humans in this region.
Gregory Abrams, thank you very much indeed.
Thank you very much, Mark, for this possibility you offered me to discuss with you. It was very interesting. Thank you very much.